So thanks, Jörn, for coming. Jörn did his PhD in Tübingen University and started studying immunology and mass spectrometry. Later, he moved to Jutland in Denmark to Aarhus University to do a tour de force with Matthias Mann in mass spectrometry and just getting the muscle to use this technique. And since 2009, he became junior professor in Freiburg University in Germany. And later in 2016, he moved to Freiburg University in Switzerland. They are quite similar names, but they are different universities. And around 2016, I contacted Jörn to interest him in plant. Because the first thing that I checked when I was looking for a collaborator with proteomics and interactomics, I went to Google Scholar and I found that Jörn has the incredible age impact of index of 47, yes, and more than 170 papers. So I strongly suggest that you Google Scholar this guy. He's going to present a little bit about interaction analysis, 2 sessions right now and later after the coffee break. And when you check the Google Scholar, the publications of Jörn, you will realize 2 things. He's an expert, he's in global, he's a monster expert in proteomics. And he has a strong policy for collaborations. He has collaborations all around the world and it's really kind to be with us today. Jörn, do you want to tell us something about protein-protein interaction? Are you familiar with this?
I'll do my best.
Yes, please.
Yeah, thanks, especially Herman and Nico for the invitation. I'm very happy to be here today. So we started talking about, or brainstorming about this workshop just before the pandemic started. And then we kept postponing, and I'm very happy that it worked out and that I can be here today. So I got the negative PCR test 1 hour before the gate opened, and I was having a nervous breakdown at Zurich airport and updating my email constantly if I will be able to catch the flight. So I'm super glad to be here. It's my first time in Argentina, and I haven't seen much yet, but I have to say I'm very impressed. It's a beautiful country, and everything is very big compared to Switzerland, where I'm coming from. So I'm really looking forward to this week here with you. I was unfortunately not here at the beginning, and I missed some of the talks. Nevertheless, I tried to coordinate with Pia and Rosario that I don't show exactly the same things. On the other hand, I think I still have to go through some basic concepts, to build up the logical story of the talk. So I hope I don't bore you too much. If it's going too much into detail, or if you're missing some information, just interrupt me, and then we can discuss this right away during the slides. So just raise your hand and let me know if I should make a break. Okay, so I think it's still important to stress again the fact, why are we doing quantitative mass spectrometry, and why is quantitative mass spectrometry so important compared to qualitative analysis? Rosario mentioned that already yesterday, that when we look at an interactome, when we want to study protein-protein interactions by mass spectrometry, we don't detect only a single protein in the sample, like when you do a Western blot, but we detect all proteins in the sample. Also, when you run a Western blot, right, I mean, your gel is full of proteins. However, you have a hopefully specific antibody, and this specific antibody will only detect the protein you are studying. But there are many, many proteins in the sample, and there are proteins in the sample which are more abundant than the protein you want to detect. However, you are blind to see those because you use only the antibody and you have the specificity of the antibody, right? In mass spectrometry, that's not the case. So when we do an IP experiment and we run that is similar, like Rosario mentioned, we get many proteins, and this is not a short list, this list continues. So we detect 500, 1,000, 100 proteins. It depends how stringent you wash, but these are really many hits. And then when you look at those, from these hundreds of proteins we detect, usually more than 90% are false positives. They are contaminants. They are binding to the beads, unspecifically binding to the antibody, and you have no real tool or way how to sort those, because the intensity that the mass spec gives back is not necessarily telling you anything about the specificity of your IP. So you need controls, you need negative controls in order to study that. So, and there are different ways how you can do that, and all of this data has to be quantitative so that you can really study the fold change enrichment, the specificity of your IP experiment. So just as a brief reminder, how do we quantitate by mass spectrometry? I have a few technical slides in the beginning, but then afterwards it will get very biological, and I will also show very many Western blots how to prove the data. This would be a mass spectrometry measurement of 125 femtomole of bovine serum albumin. That's like a standard protein we use to calibrate the instrument on our instrument. So this would be the chromatogram. So we have the separation over time, and here there would be the relative intensity. And all these little peaks here that you see, those would be peptides from this 125 femtomole of bovine serum albumin. So per definition, if we start with 125 femtomole of a protein and we chop that up into peptides, all of the peptides should be present with 125 femtomole, right? So it should be the same amount of peptides. And now just as an example, this would be two different peptides. This would be the mass chromatogram of these peptides. So here we would have the mass of the first peptide, that would be 751.8, so mass to charge ratio, and here 417.2 mass to charge ratio. And then due to the fragmentation of the mass spectrometer, we can also say which peptides these are. They have different charge states, but both of these peptides should be present with 125 femtomole. So I know it's a bit hard to read, but we have here the intensity of the peptides. And when you look into this intensity of the peptides, you would see that this peptide is 3 times more intense than this peptide. So if you compared these peptides and you would look at the fold change, you would see, this guy, I have 3 times higher intensity, so you might deduce I have 3 times more of this peptide in my sample. But that's not the case, right? We start with 125 femtomole, and this peptide is exactly as abundant as this peptide, but the intensity you get back from the mass spectrometer is a completely different intensity, right? So in order to make sense out of your data, you have to come up with quantification methods, how you can study these peptides, right? Why do we have this different intensity? So first of all, in order to measure a peptide, we have to ionize it and transfer it into the gas phase to bring it into the mass spectrometer. And these peptide ionization properties are very different from peptide to peptide, and it's super hard to predict those. So that is gas phase chemistry. There's no good algorithm where you could type in your peptide sequence and then it would tell you how well this peptide ionizes, right? So you cannot solve this problem, these different ionization properties, on a computational basis. You have to solve this problem on an experimental basis. On top of that, because we're doing bottom-up proteomics, right, so we digest our proteins into peptides, we use usually trypsin as an enzyme. Trypsin cleaves with a different specificity. If we have some secondary structures or tertiary structures, even depending on the lysis buffer you are using, the accessibility of trypsin might be different. So therefore we might not generate the same amount of all of these peptides, right? And therefore you cannot discriminate, or you cannot directly compare the intensity of the mass spectrometry for 2 different peptides. However, what you can compare is the peptide A in sample 1 and peptide A in sample 2. So we can perform these relative quantification approaches. And this is basically what we always do when we talk about quantitative mass spectrometry. We use this relative quantification, comparing exactly the same peptide in different samples. This can be by stable isotope labeling, what already has been introduced, or it can be label-free. Regardless what you do, you always compare the same peptide in 2 different samples, in 3 different samples, and so forth. Either label-free or by using labels. And just to quickly, briefly summarize again the different approaches. We have metabolic labeling, we have chemical labeling, we can spike in the standard, and we have these label-free approaches. I don't want to go into all the details because these have already been introduced. I just would like to highlight that each of this method has a different weakness and a different strength, right? For example, when you want to study human samples from the clinic, metabolic labeling is not a real option, right? So you use the chemical labeling approaches, for example. These chemical labeling approaches you can also multiplex quite extensively. You can compare, for example, with the newest type of reagent, 16 different samples in one mass spec measurement, right? So if you have many, many samples, this chemical labeling is a very good strategy. What I would like to mention, because we are doing that a lot, and also will spend a bit more time on that, is the Silac labeling. This is this metabolic labeling here. So here you use the metabolism of the cell to incorporate the label. And the nice thing about the Silac labeling is what you have shown here in this figure is that you can combine your sample already at the cell level, right? Because the entire proteome has been labeled, and you can discriminate the proteome of your different samples, even if you combine at the cell level, and then your protein biochemistry, your IP, your whatever you are doing, you can do this in one tube, which means that the unspecific losses you have doing your purification procedure account to the same extent for both types of your samples, right? Therefore, Silac labeling is very accurate compared to other labeling approaches. Because in the chemical labeling approach, you can do that on the protein or on the peptide level, but depending where you do, you still treat your samples in different tubes, right? And you might have unspecific losses, which, because they're unspecific, you don't know them, right? And you only will see this later than when you do the mass spec analysis. When you do the spike standards, when you synthesize the peptide, this is very difficult to do on a proteome scale, right? Because you have to synthesize all your peptides. So this you can only do for single experiments, usually. And then the label-free approach is quite cheap, and the new algorithms are very robust, so it works really, really well. But still, it's rather inaccurate compared to the other approaches. So whereas with the metabolic labeling, depending on the setup, maybe you can already say that the 1.5-fold difference is statistically significant. With the label-free approaches, you commonly need at least like a 2 or 3-fold difference in order to be able to say that this is a truly significant difference, because you have these unspecific losses during the purification procedure. So this is just something to keep in mind when you start to design your experiment, that you are aware that we have these different approaches, and then you have different arguments why you want to use one approach or the other. One is of course accuracy, but another can also be amount of time you spent on the mass spec, amount of money you want to spend on different parts of the experiment. So this is everything one should keep in mind, and also something one should discuss with the experts before you start, because they might have a good idea and could say, oh, did you think about this? You know, maybe chemical labeling would be an alternative for you, and then we don't have to run 20 samples or 16 samples. We can do this all in one measurement by mass spec, right? So we are doing a lot of metabolic labeling, I mentioned that, and we are using Silac. This is stable isotope labeling by amino acids in cell culture. And just briefly, basically you grow your cells in these different isotope labeled media, and then you do your standard proteomics experiment. And if you measured those samples one by one, you would get one signal for your light peptide and another signal for your heavy peptide. So the nice thing now is that you can combine these samples and you can measure these in a single mass spec measurement. This makes also the quantification more accurate. And then you get basically these duplets in the mass spec, and you can relatively quantify the abundance of these different peptides. So for example, here we would have a ratio of 1:1. You would expect, if you don't perturb your sample, that all peptides should be present in a 1:1 ratio. would be like a control. But of course, it gets interesting when you start to perturb your sample. For example, we could imagine that we treat sample A with a growth factor, and we look at phosphorylation events that happen, right? So we treat the cells with a growth factor, some kinases get activated, they start phosphorylating, and we have these phosphopeptides which get more abundant, right? And then it would look something like this. We have a higher abundance of the light peptide compared to the heavy peptide, and then with this we could basically study signal transduction inside the cell. So what kind of amino acids do we use? You know, there are different characteristics you have to think about. So usually these should be essential amino acids so that you don't have a label spread, that they are not getting metabolized in the cell. Each peptide should hopefully have a label and be quantifiable. And also the pattern of the label should not be too complex so that from the bioinformatics approach, you can easily sort out the different labels. That is what makes plant labeling with the metabolic labeling so complicated, because in plants they reshuffle everything and then you have a dilution of your label in many different variants, and metabolic labeling of plants is very complicated also from the bioinformatics point of view to make sense out of the data. So therefore, when you do mammalian cell culture, so we do most of our experiments in mammalian cell culture, we use arginine and lysine. Lysine is essential and arginine is basically conditionally essential. When you provide enough arginine in the medium, they usually only use the arginine of the medium. Because we use trypsin as a protease, each peptide has a lysine and an arginine residue except the C2 so we can quantify all of these peptides then. And then in general, each peptide should contain only a single labeled amino acid if we don't have miscleaved peptides with trypsin. But in general, it works really well. And then what we're using stable isotopes. So we use 13C and 15N to label our amino acids. So for example, we would have this amino acid arginine 10. This amino acid is 10 Dalton heavier than the regular amino acid arginine, right? Because we introduce 6 carbon 13 atoms and 15 nitrogen 15 atoms. The counterpart from arginine labeling would be lysine 8, and here again we use 13C and 15N labeled lysine. So these are stable isotopes, have nothing to do with radioactivity. You just buy them and can use them in any lab, so it's not a problem. So when you grow your cells, you usually have to keep them in the Silac medium for at least 5 to 6 cell doublings, and this would be here an example, you know, where we from the original publication from the group of Matthias Mann, where you keep the cells for different amounts in these labeled amino acids, and you can see that after 12 hours you already have like almost 50% of your protein labeled. So these are usually cancer cell lines, right? So they have a very high metabolism, but you still, in order to get the complete labeling of the cell, you usually have to wait a couple of days. Here an example where we have 5 days, keeping in mind that these cells replicate every 24 hours, right? So we have something like 5 cell doublings, and then you don't see the non-labeled peptide anymore. Your proteome would be completely labeled, and then you can perform your biochemical experiments. So you have to keep in mind that you first have to label the cells before you can do your biochemistry. And then it's also good to check incorporation. So here we looked at the incorporation of the arginine label and of the lysine label, and then you can see that after 5 to 6 cell doublings we have close to 100% label incorporation. We never get 100% label incorporation, even if we keep it for half a year. And why? Because the reagents that we can buy are not 100% pure, right? So when we buy the labeled amino acids, we get them with a purity of 98%. So then also, of course, you only get 90% of label incorporation. But this is not a problem, because usually the accuracy of the mass spectrometer, the technical accuracy of the mass spectrometer is something between, you have an error between 10 to 20%. So with this 2% having this not labeled is basically in the noise of your mass spec measurements, right? So you don't have to worry that this could be not accurate enough. So Silac is super versatile. You can make many different types of experiments with this. Usually we can compare 3 different conditions, and what is very well known is this kinetic Silac approach, where you would have your 3 different labels, and then you treat them with a growth factor, for example, for 10 minutes, 20 minutes, and then you look changes on phosphorylation dynamics. You can do treatments over over hours or days to look at changes on the proteome scale, and then you can basically perform kinetic analyses and see how proteins accumulate, how proteins change their phosphorylation state, and so forth. However, you can also do classical pulse or pulse chase experiments, and I would like to spend a little bit time on that, because this is something that you cannot do or not well do with chemical labeling. But with Silac labeling, you can do these pulse chase analyses to look at protein turnover, to study protein synthesis, protein degradation, and this might be interesting for some of you. And then last but not least, you can also do the spatial approach to study organellar proteomes. This looks very complicated, but in the second hour of today, I would explain you how you can perform organellar proteomics using this spatial Silac labeling. Okay, but just as a brief slide in, so to say, how can you use mass spectrometry to look at protein turnover. So classically, this was done with radioisotope labeling, right? So this would be an example where people used 35 sulfur labeled methionine, and you do a pulse labeling over time, and you look at the incorporation of your radio labeled amino acid, right? So you can do this with a pulse, or you can do this with a pulse chase to look at the loss of the label, right? With a pulse, you would study protein synthesis and protein turnover. With the pulse chase, because you study the loss of the label, you would study protein degradation. So exactly the same what you can do with radioisotope labeling, you can do with stable isotope labeling and couple that to mass spec. And whereas here you get bulk data, right? So you get degradation of the proteome, right? With the mass spectrometry, you get degradation or turnover rates for each single protein you are studying, right? So it's a completely other data completeness that we generate. So if you want to study protein turnover, you do these pulse labeling experiments. And now I'd like to just get the vocabulary right. Protein turnover with the proteomics view is not the same as protein degradation, right? Protein turnover would be the sum of protein synthesis and protein degradation. And when we want to talk about turnover, basically we have to keep in mind that we talk about synthesis and degradation instead of talking about degradation only or synthesis only, right? So here in this example, we would have a heavy labeled culture. We do this light labeling in a pulse. This can have different times. And then usually you get these two signals. That would be our heavy peptide, and that would be our light peptide. And now you see here that the heavy peptide during this pulse can only get less, so that would be the degradation, the loss of the heavy peptide. The light peptide gets synthesized, so this would be the synthesis of the heavy peptide, of the light peptide, sorry. It can also get little bit degraded, and then the difference would be the protein turnover. And these are quite simple experiments that you can perform in order to look at the dynamics of your proteome. If you want to specifically study protein synthesis, this is also possible, but for this you would need three labels. So here you would start with a non-labeled cell culture, and then you do this pulse labeling in the medium heavy and in the heavy label, and then you can compare protein synthesis of these two labels, right? You don't care about this light peak, because the light peak comes from both cell cultures, but you would study your accumulation over a given time point. So this would be 24 hours, for example. And then you could see that in this example, within these 24 hours pulse labeling period, the heavy peptide has been more synthesized than the medium heavy peptide. And you can look at relative changes in protein synthesis, right? You can also do pulse chase experiments to look at protein degradation. So here again, you would need three labels. You have the light label, then you have a pulse in heavy and a pulse in the medium label. And then you do this chase period again in the light label, and that would tell you the differential degradation during this chase period. Because again, like in the other example, we don't care about this light peptide, but we look at the loss of the label comparing the medium heavy to the heavy peptide, and then we can study changes in protein degradation over time. All right, but the actual main topic of the talk, right, would be how to study protein interactions. And I think here it's important to mention that we have two ways to study those. We can purify proteins of interest, or we can do a protein correlation profiling of proteins of interest. And I will talk about both examples, right? So we will talk about, now in the first hour, we will talk about the affinity purification and a bit about proximity labeling, although this has already been discussed in detail yesterday. I would just like to give you some biological examples, how it looks like. And then about protein correlation profiling, we will talk in the second hour. So the underlying concept is that in the one, you do an IP type of experiment, and you enrich your protein complex or your organelle that you want to study. And in this protein correlation profiling, you don't try to enrich, right? You basically measure the relative distribution of proteins over a biochemical fractionation experiment. And by following this relative distribution over a biochemical fractionation experiment, you can also deduce which proteins are at least in a similar compartment. You know, it might not be protein-protein interaction, but you could study, for example, the composition of an organelle, also the composition of a protein complex, depending how it behaves. But I think it will get clearer when we discuss this then in detail. If you start with your protein of interest, it's important to basically be aware of what is already known in the literature. I mean, this was mentioned, right? So it's always a bad idea to just do an omics experiment and then hope that the data will tell you something, right? So you should still basically do your homework and read what is known. And for example, when you study protein-protein interactions, you have to be aware that we can have very strong interactions where we have dissociation constants in the nanomolar range. We can have very weak interactions where we have dissociation constants in the micromolar range. And depending on what type of interaction you might be interested in, you might have to use a different biochemistry, right? So from a chemical point of view, we can discriminate this obligate or permanent interactions from this non-obligate or transient interactions. And these transient interactions can be strong or weak. And for example, when you are interested in these very weak transient interactions, then proximity labeling might be the method of choice, because you will not be able to enrich the proteins by a classical affinity purification. But when you know that you have a strong interaction, then affinity purification might be the method of choice. These obligate or permanent interactions, those you should always get. Those would be like the positives controls you could use in your experiment, right? And then of course we can have homo-oligomers or hetero-oligomers, and commonly from the biological point of view, we are interested in multi-protein complexes. So this would be hetero-oligomers, many different subunits, and you want to study how they are regulated and how they are changing their interactions. So in the classical affinity purification, we can do an endogenous affinity purification. We can use a single tag, or people also use a double tag purification. And again, right, you have to know your method and you have to decide which strategy you want to follow. If you use the endogenous purification, this is very nice from that perspective that you don't overexpress, right? So you don't perturb the cellular network. However, you are very much dependent on your antibody, right? You need a good antibody to be able to perform a good IP. And I would always recommend to test your antibody first in a Western blot and then go to the mass spec facility. Because if your Western blot looks like crap, very likely the mass spec data will look like crap, right? So you can do these tests beforehand before you decide to perform a rather expensive mass spectrometry experiment. If your Western blot looks very clean and you can nicely enrich your protein and you get a very nice pattern, right, then very likely the mass spectrometry data will look very nice and you will get many more proteins identified, right? So we work a lot with these single tags. Of course, you overexpress, but usually from a proteomics or mass spec point of view, this does not interfere too much. And then you can use the antibody against the tag and then IP the entire complex. Nowadays with CRISPR-Cas, you can also genomically tag, right? So then you would not have this overexpression problem anymore, but you would genomically tag your protein of interest and then could still use the efficient tag purification in order to get your interacting proteins. And then especially in yeast, people used a lot this dual tag. You have this tub tag purification procedure where you use two different tags. One can, the first IP, you would use protease, for example, TEF protease to cleave the tag, and then use a second tag to get a very clean complex. You can still do that, but from a mass spectrometry point of view, that would not be really necessary because we don't care too much about the contaminations. I will show you examples where we can use basically the contaminations as a statistical background, so they actually help us to interpret the data. So I would argue you don't need a very clean purification, but you need a very good enrichment, right? So you can skip one washing step, but you want to have an antibody that really works well in the IP, right? So, and then what we have heard also yesterday, the negative controls are really absolutely crucial, right? Without the proper negative controls, you will not be able to interpret the data. For this, we need quantitative mass spec, right? And so the worst case, I mean, it's still okay, right? We also do that, but just from a data interpretation point of view, it's really the worst case. If you do an endogenous IP, if you cannot do anything else, you use the IgG control antibody, right? Why is that the worst case? Because we have a lot of, we have unspecific binding to the matrix, meaning to the beads where you couple the antibodies to, but we also have unspecific binding to the antibody itself. So if you use the IgG control antibody, that's a different antibody than your actual antibody, and then you will not be able to discriminate true binders from different non-specific binders due to the fact that you used another antibody, right? So this can make data interpretation a little bit more complicated. So this would be the worst case. If you do an endogenous IP, usually what works much better and what we do a lot would be then do a knockdown or generate knockout cells so that you use exactly the same beads, exactly the same antibody in the knockdown or knockout, and then you can compare this one-to-one to your original IP. If you use a tag, of course, this makes it much easier. Then you just use cells which you don't, where the cell, where you don't have cells expressing your tag variant. If you are scared about clonal selection, if you have different cell lines that might change their characteristic, you could also use here inducible expression systems that you induce, for example, with doxycycline, your protein of interest with the tag only for 48 hours, and then you compare the different IPs. Yesterday, Rosario said that you have to be aware that the proteome can differ, and that is also here the case, right? If you have differences in the proteome, those might obscure your data a bit, but you can measure the proteome and you can try to determine those differences, right? And then if you see that the proteome is very different, that you have very much more of one protein in one cell type compared to the other, this would help you to say, okay, if this protein is now one of our binders, maybe it's not a binder, maybe it's an unspecific interaction and it just is more in the sample because it's more in the specific cell type, right? So you can also try to address that problem experimentally. Okay, so one of the first papers that came out also from the group of Matthias was this quantitative immunoprecipitation compared with knockdown. So here, that was basically before CRISPR-Cas, so here they used as hRNA-based knockdown to have your knockdown cells and your wild type cells, and in this example you have again a Silac labeling approach, so you use two different Silac labels, and here I just showed it because it nicely highlights the different non-specific binders that we have. So the green protein would be our bait, right? So the green protein we are missing in this example because we did the knockdown, so we miss our bait and we miss all the green specific interaction partners of our knockdown. However, we have here in red the non-specific binders which bind non-specifically to the beads, right? And we have also a cross-reactivity of the antibody, which is the same because your antibody might cross-react with something else, right? You are not aware of that, But if you compare now the knockdown cells compared to the wild type, you will be able to discriminate all this, because only your true bait and the interaction partners of this bait should be higher enriched, the non-specific binders to the beads, and also the cross-reactivity of your antibody should not interfere with data interpretation. And just to give you a flavor how the data could look like, right? So here they looked at the enrichment of the Sybil. This is a E3 ligase, which is important in growth factor signaling. And you can see all the proteins they identified. I didn't count the dots, but I guess these are thousands, right? And then you have here, dependent on the statistical parameters you use, you have something like 5 interaction partners that they believe, right? So you really need these controls and the quantitative data to look at that. I also would like to mention that statistics are important, but you also have to use your brain to look at the data. Don't just look at the p-value, right? Because very often we have proteins which are known interaction partners and that we enrich, which don't have then a p-value of 0.05, but of 0.055 or of 0.06. And then very often when we discuss with colleagues, they just look at the ones, you know, that are marked as statistically significant, and then on the rest they don't even look at that data, right? And that is a mistake, right? You always have to look at the full list, and then when you look at this graphically, right, also the graphics will tell you something. So here only these proteins were determined as statistically significant. But looking at the data, you can say, okay, these 3 proteins are very likely true interactors. Maybe even these 3 proteins or these 5 proteins are true interactors, and then you have the big noise level, right? So it's important to do the statistics, but continue to go down the list, look at the distribution in a graphical point of view in order to be able to get a better feeling of your data, to know what is a true interactor, what could be a transient interactor, a true transient interactor, and that's why the enrichment did not work as well as for the more stable interactors, right? It's all hidden in the data, but you have to spend enough time on your data in order to see these differences. Okay, this would be an example from our group. Here we studied the interactions of collagen 7. Collagen 7 is an important extracellular matrix protein, and we have a group of diseases, human skin diseases, where we have a mutation in collagen 7, and the patients suffering from this disease, they don't have any collagen 7 anymore. And we wanted to know the protein-protein interactions. So in this setting, we would have our wild type cells. So those would be cells from healthy donors which express collagen 7. And we have cells from our patients which are basically like collagen 7 knockout cells, right? So what you can do here then, you use you do an IP against the endogenous protein, so that would be the same antibody, right? And then here you would purify collagen 7 and all its interaction partners, and this would be our negative control. Here we would not purify collagen 7 because these are the knockout cells, and this would only be the non-specific binders. So we did this again in a Silac setting, and you can do this experiment in 2 ways. You can do 2 separate IPs, how it's shown here, right? So you have your light labeled and your heavy labeled cells. You lyse them, and then you do 2 separate IPs, and then you mix on the elution step of the IP, right? So with these 2 separate IPs, you will get stable and transient interaction partners because you mix at the elution step. You can do this experiment in a different way because you have already metabolically labeled cells, right? So the entire proteome of your cells have already been labeled. So you can already mix the cells at the lysis level. You can basically mix your cells, lyse them together, and then perform the IP. If you do this, you only get the stable interaction partners, because the transient interaction partners, because you have it combined on the lysate level, they bind and let go, bind and let go, right? So the transient interaction partners are lost, and only stable interactions are preserved. And depending on your protein, this might work well or not. You have to be a bit lucky. In our case, it worked super nice. So here we would have, these are Silac ratios. It's always compared to the knockout cells, right? It would be our wild type cells compared to the knockout cells of the combined IPs. And here we have the wild type cells compared to the knockout cells of the endogenous IPs. And here in blue, this would be the background, right? So that's why I mentioned the background usually is not an issue. It helps you to normalize the data. So everything in blue are non-specific binders. And now we have binders here in red, which we get in both settings. We get them in the combined IP and in the separate IP. Those proteins that we get in both settings, those are the stable interaction partners, because regardless when we mix the lysates, we always enrich the same proteins. But then we have a couple of proteins which we only got in the separate IPs, right? So we only got them significantly enriched in the separate IPs. You can see here in the combined IPs, they're basically at the same enrichment level as the noise, right? So these proteins, per definition, would be transient interaction partners, because when we mixed the lysates, we could not enrich them anymore, right? So then you can put that together. We have here collagen 7. So this would be our stable interaction partners, and this would be our transient interaction partners. And for this type of data, it made a lot of sense, because the stable interaction partners are mostly proteins which help in the maturation process of collagen 7, whereas the transient interaction partners, those were mostly cytoskeletal and vesicle proteins which helped in the secretion of the collagen 7, right? So depending on your protein, you might learn something about the biochemistry of your protein. So this is something which is one could keep in mind. So this was all affinity purification based experiments. So now towards the end, I would just like to give you one example of bio-ID data, of proximity labeling, what Rosario mentioned yesterday already. So we are working a lot on autophagy and on autophagy signaling, and especially in Saturday I will tell you about the signal transduction dynamics in autophagy signaling. But we are very much interested in this protein kinase complex ULK1. ULK1 is the most proximal kinase complex important for autophagy regulation. It's the human autolog of yeast ATG1, and it's important for autophagosome biogenesis. So in autophagy, you form this double membrane, in macroautophagy, you form this double membraned organelle, which basically enwraps cytoplasm and targets it to the vacuole or lysosome for degradation. And this ULK1 is a heteromeric complex. We have ULK1, which is the catalytic subunit. We have ATG101, ATG13, and FIB200. So these 4 subunits are important for the complex to be functional. It gets recruited to the endoplasmatic reticulum and then phosphorylates many target proteins in order to start this autophagosome biogenesis at the endoplasmatic reticulum and in order to form the structure. I will talk about this more in detail on Saturday. Basically now it's just important that we wanted to study the interactome and the neighborhood of this ULK1 protein complex. So when we did the interactome, we got something like 10 binding partners for each of these 4 subunits. When we did proximity labeling, this was the network that we got with proximity labeling. So it's really a very good alternative approach, right? It's not that the one is better than the other. If you can always do both, because you will get completely different data sets if you do the affinity purification and if you do this proximity labeling. So here we have the 4 proteins, ULK1, I cannot read those, right? But basically it's ULK1 and the others, right? And now you get the neighborhoods plus the binding partners. And this would be, you can compare this almost to an imaging approach, because by the neighborhood you will basically learn, is this protein more at the ER, is this protein more at the Golgi, is this protein more at the plasma membrane, and it's a very different type of data you generate. And for example, then you can put that into string database or some other software for protein-protein interactions. You get the Go terms, and then by the Go terms you will learn that at what kind of organelle your protein is localizing to, and so forth, right? So we do both usually on a routine basis, because it's a complementary set of data. You have to be aware of that, dependent on which side of your protein you add your either peroxidase or your biotin ligase, you will get different data, right? Because it's really the case that you label in a surrounding of 10 nanometers, and depending how large your protein is or of the structure of your protein, you get different proteins enriched. We saw that, so if you can do both. And this is just to keep in mind, I mean, this was already mentioned yesterday, that of course what is very different from these different proximity ligation approaches, if you use the peroxidases or the biotin ligases, is the amount of time you need for labeling. With the peroxidases, you label minutes. With the ligases, depending on if you do turbo ID or not, you label a couple of minutes to hours. And then also the amino acids that get labeled are different. All right, this would be the last slide for this. Sorry, I thought this was a summary already. This was the last slide for the different types of negative control for this proximity ligation. This was also already covered yesterday. I would like to mention that in our case, we usually prefer to use one of these three, right? So either we fuse to an unrelated protein such as GFP. This is not perfect, but still it works okay. We have an inactive variant or mutant variant of our bait, or we have a protein which goes to the same location. This is a bit a question of taste, you know, what you prefer or not. But with these two controls, we at least in our case, we had a bit of a problem. The problem is if you use the free enzyme, again, now in our hands, is much more active than the enzyme coupled to a bait, right? So if you have the free enzyme, you have very strong biotinylation reactions in the cell, and you enrich many, many proteins. So if you use this as a negative control, we had the impression that we had too many false negatives, because the enzyme is so active, you have many false negatives. When you use no enzyme at all, you have many false positives, because then basically your enzyme, your fused enzyme also has noise, right? And you have many false positives. So with these three controls, we thought that we got the cleanest data, that it's a good compromise between false negatives and false positives, and the data looked most sensible. All right, so now I'm at the summary. So in the beginning, we talked about the ion intensity and why we need these quantitative strategies to study protein-protein interactions. Only chemically identical peptides can be compared by mass spectrometry based on their intensity. So therefore, this stable isotope labeling does not interfere with the ionization properties of the peptides. So therefore, these labeling approaches are usually using stable isotope labeling. Silac-based affinity purification mass spectrometry can help you to discriminate stable and transient interaction partners. That's what we discussed with the collagen 7. Depending when you when you mix your lysate, you can discriminate that. And proximity ligation also works really nice in our hands, and it's more like an alternative to organellar proteomics or even to imaging approaches to ask, you know, where does your protein localize in the cell. Regardless what you do, you always need these very good negative controls, and it's worthwhile to spend enough time thinking what would be a good negative control. This will make your life much easier in interpreting your data. Because if you do proteomics experiment, the good part is you will always have data. So you can always make some figures and speculate. So it's not the problem that you don't have enough data, it's rather the opposite. You have too much data, and it's really difficult to make sense out of your data. And you do need these negative controls in order to make sense out of your data. So this would be the end for the first lecture.
Thanks, Jörn. Really, really amazing talk. I have two questions first. Sorry. Silac, could you apply it to yeast? Silac based?
Yes, so we do a lot of Silac in yeast, so we use then lysine arginine auxotroph yeast. You can also use the wild type yeast, but then you have to increase the concentration of amino acids in the medium so that they then don't make it themselves. That gets quite costly from the Silac labeling, but we have very nice data when we use lysine arginine auxotroph yeast strains, and then if you have the proper negative controls, it's not a problem for yeast. We also use now a lot of proximity labeling in yeast, because there was also a problem because the biotin levels are high, but we have very nice turbo ID or mini turbo ID data from yeast. It works really beautiful. We have all the protocols and we are happy to share those.
And one more, actually I had much more than one. Let's later for later.
Yeah.
Could you back to the previous slide, when you take negative controls, you say the ligase, the biotin ligase for proximity labeling, the enzyme, the free enzyme mark a lot.
Yes.
Could you target to the organelle? Like for example, UL key, if you use proximity label, could you anchor the proximity label to the membrane just to make a much strict negative control?
Yes. I guess yes. So the problem in our case was that we are always studying cytosolic protein complexes. So therefore our negative control was really the free enzyme. But if you are targeting that to a membrane with a membrane anchor, I would expect that reduces its activity, and then it would be, that would be similar then to a protein that goes to the same compartment, right? But in our case, I didn't mention that, but for the cytosolic proteins, we got much better data with the bio-ID data than with the Apex. If you also go into the literature, it seems that the Apex labeling works really nice at membranes or at membrane-bound compartments. But if you do this in the cytosol, at least in our hands, we had really many false positives. So too much noise. Yes.
Thanks a lot. Vielen Dank. Any other question? Yeah.
Congratulations for your presentation.
Thanks.
I have two questions. When you mentioned label strategies, if I have many amino acids labeled or protein labeled, could you have any problem in relation to the retention time between heavy and labeled and unlabeled?
So
in relation to the ionization from different ionization from different label.
Yeah, so 13C and 15N usually has no influence on retention time. When you use deuterium labeling, deuterium has an influence on retention time, and depending how many deuterium atoms you use for labeling, that can be a problem. I actually have a slide for that. So here you see that if you use one deuterium or two deuteriums, you still have no issue in retention time. But here you compare 3 deuterium to 3 hydrogen, and you see that the peak is already split. And then if you have 4, then it's even more split. So dependent then where in the peptide you introduce the label, it's also different if you have the labeling of the termini or internally. Deuterium can cause a problem, and you would have to take care of that for the quantification. If you look at carbon 13 or nitrogen 15, they usually don't have a shift in C18 retention time, and that's not a problem.
Okay, and the other, when you mentioned that is important, my question is, it's mandatory a high efficiency for trypsin digestion, or depend on the limit of detection?
Exactly, right. I mean, why are we using trypsin? Trypsin cleaves after arginine and lysine, and arginine and lysine, due to their basic side chains, are positively charged when you use acidic buffer conditions. So that's why we're using trypsin, because then this supports the ionization of the peptides. But dependent on your protein, trypsin might not be the good protease, right? We have other. So if your peptide, if you generate a too short peptide, meaning less than 6 amino acids, you cannot really discriminate that. And if trypsin generates too big peptides, like more than 30, 40 amino acids, also you cannot really measure that in a standard bottom-up shotgun proteomics type of experiment. So when we are studying a specific protein and we see that the tryptic peptides don't cover the amino acids we are interested in, we use different proteases, right? So you can use ASP-N, GLU-C. We also use elastase or thermolysin, which are more non-specific proteases. And then with those, you might be able to have a better sequence coverage. But otherwise, of course, the better the protease activity, the more peptides you generated, the less starting material you need, right? Okay. If you say you have problems with trypsin efficiency, maybe you could use more starting material or something. So in general, I think for us, it was never a big problem. I'm not sure if this answers your question.
Okay, thank you very much.
Yeah.
One more question? Yeah, Elvia.
Hi, great talk. I have a question about the medium used for the Silac labeling. Those are defined mediums or
So basically, this is like shelfware. You can buy that, right? So basically, we use mostly DMEM medium, right? And then you buy DMEM medium without arginine and lysine, and then you just add your arginine and lysine to the medium, right? But you can also use different combinations of amino acids. You have also RPMI. So the common media are shelfware. You can just order that at the different companies. If you have a very specific medium, you can also order that, but then this is custom made, and then it gets a bit more expensive. You have to be aware of that. You have to use dialyzed FPS because in the FPS you also have a lot of amino acids, right? So then basically you would not have proper labeling, but you can basically buy dialyzed FPS, and then you buy this arginine lysine-free medium, and then you just mix it, and it's very straightforward.
Okay, yeah, my problem is that I work with Trypanosoma cruzi, which the medium is not defined, in which you grow them.
Okay.
It has a liver infusion.
Okay.
I don't know what you suggest for that.
So then, so for sure, what you would have to do is dialysis to get rid of the internal amino acids, right? But then you get rid of all amino acids, right? So then you would have to add all of the amino acids plus your Silac labeled amino acids, right? So there you would have to really set that up. That might be you might need a couple of months to get the correct composition.
Okay.
Yeah.
And the serum, the bovine serum.
The bovine serum you just buy, again, right? So it
has no amino acids.
So it's I think they use commonly like a 3 kD cutoff, right? So everything smaller than 3000 Dalton is dialyzed out, but all the other things are still inside, meaning also the growth factors are still in. The cells grow a little bit slower, but not dramatically slower. And we use that for cancer cell lines, but also for primary cells. So we use skin fibroblasts, keratinocytes. They all grow fine in this medium. So so that one could definitely try.
Okay, thank you. And I will present my work about proximity labeling on this organism. If you can give me some
yeah,
advice,
I'm happy to give you input. Yeah, I'm here the entire week. Yeah.
Thanks.
Yeah.
Super, super. Thanks, Elvio. Any more?
Thank you, Jörn, for the beautiful presentation. A couple of questions. When you are combining the lysates, what type of control do you do beforehand that you are mixing? You have the same amount of protein. Do you normalize to cell counts? Do you measure protein content?
We always normalize on protein content.
Protein content.
So yes, we always normalize on protein content. I mean, in theory, in the old days, we also just did cell count and just looked at the pellet so the eye works surprisingly well if you have a similar sized pellet and the lysis works similar well. That usually is okay. But then also what we often do is that before we do the IP, we take like 1% or 10% of the lysate and just measure that one to know exactly the mixing ratio. And then you directly see I mean, if you have a mixing ratio between 0.8 and 1.2, that is all okay, right? If you have a mixing ratio of 2 or so, then the quantification gets less accurate.
Which method do you use? Bradford, 280?
So we use usually the BCA method. Bradford we use. So the BCA method works is less detergent sensitive, right? So then we usually use BCA. If we have detergent-free samples, you could also use the Bradford.
Okay.
Yeah.
And finally, which software do you prefer to analyze your data?
So we do everything with MaxQuant and Perseus.
Okay.
I mean, but I think it's a bit like historically, right? Because I did my postdoc with Matthias, and then you know how this developed, and then you follow this path. I mean, but we are very happy with MaxQuant and Perseus. And all the data I showed was all on thermo machines. It's HFX or the QE Plus, or even the old Orbitrap XL. So it's all Orbitrap based and all MaxQuant Perseus based.
Thank you.
Last question? No. Well, thanks a lot.
Thank you.
And we are going for the coffee break. But before everybody get up, stand, people that need to give the flash talk, go to Rodrigo to give the presentations. Many thanks.